So now in this video lecture we will study about the extraction of nucleic acids, isolation and extraction of nucleic acids purifications. Now what is the purpose of doing this nucleic acid purifications? Why we do nucleic acid purifications? The purpose of doing nucleic acid purifications is that DNA is extracted for the downstream application ranging from PCR, electrophoresis, sequencing, fingerprinting, and cloning. DNA extraction removes protein, nucleases, and other impurities. So that's why DNA extraction or nucleic acid extraction is important. Now we will see different methods for the DNA extraction. First one is thermal extraction, then aqueous solution based extraction, organic solvent based, solid phase, and solid phase isolation using the magnetic pits and columns. So these are some of the methods. Now we will see that what are the outlines of an aqueous solution based DNA extraction method. First one is breakdown of the cell wall, then centrifugation, precipitation, and we will proceed for the elution step. Now we will discuss the first, that is thermal extraction method. Thermal extraction is quick, crude, and low cost. It can be used to extract DNA from pure bacteria. Sample is heated at 100 degrees Celsius for 5 to 15 minutes. That's simple. And now we'll see aqueous solution extraction. So in aqueous solution extraction, there is lysis, resuspension, there is lysis with EDTA and all, and then is separation of the phases. The steps involved in the aqueous phase of extraction is breaking cell open to release nucleic acid into lysate. The first step of nucleic acid extraction process is cell lysis, that is mechanical or chemical by chelating with EDTA or with lysis buffer. Second is separating the DNA, and then third is centrifugation. The aqueous phase contains the purified DNA. Now we will see precipitation the DNA with an alcohol, and fifth step is drying and desuspending the DNA. The nucleic acid pile is allowed to dry before desuspension. Now dry and desuspending the pellet. The precipitating the DNA with alcohol is important, and confirming the presence of the DNA by electrophoresis. Now organic, we have discussed organic. Now we'll discuss the organic phase extraction with phenol chloroform extraction. So these are some of the basic steps of the nucleic acid purifications. Solid phase extraction we use different kind of columns like silica phase. So nucleic acid basically bind with the silica column, and there is a washing step initially, and the elution will be done in the TE buffer or nucleus free water. So these columns are commercially available. Now another step is important, that is PCR fragment cleanup. There are different kind of PCR cleanup kits are available in the market. So PCR fragment cleanup using spin column purification method, as you can see in the figure, can be used for purifying PCR product and other molecules greater than 200 base pair. Now you'll see PCR fragment cleanup using XO-SAP-IT reagent. So this is reagent also used in the. So now you'll see the mitochondria DNA isolations. So mitochondria, these are several steps that need to be carried out isolating, that is tissue homogenization, isolation of mitochondria, removal of genomic DNA, gDNA, and mitochondrial DNA isolations. Now we will see total, what is RNA extraction, total RNA purification, organic RNA extraction method, triazole method, magnetic bead method, silica membrane method, mRNA purification method with oligo-DT. Different methods of RNA extraction are triazole, magnetic bead, and silica-based method. Now we'll see RNA isolated with guanidinium isothiocyanate. Phenol chloroform extraction is generally DNA and protein free, and it can be used for the RT-PCR also. RNA extraction using guanidinium isothiocyanate phenol chloroform. Step 1. Sample of interest is treated with monophasic phenol. Step 2. Sample is subjected to organic extraction. Step 4. The RNA can be removed by precipitation from aqueous phase. Now you'll see these are the different steps you can see and note down. Now types of RNA: messenger, ribosomal. As you know, extraction of RNA using the organic reagent. This is traditional method based on pH, that is pH 4 and pH 8. Now magnetic based method for high throughput purification of the RNA. So messenger RNA basically present on bead or column, combines with the poly A. The so-called charge method, pH less than equal to 6.5, magnetic beads bind. And there is silica membrane based RNA purification method also. Here we use the commercial column. Filter column purification can be used to purify RNA by adjusting the pH or salt of the solution. The popular RNA isolation method is compatible with vacuum also. RNA isolated using column purification is suitable for the Western blot. Isolation of microRNA and small interfering RNA. So isothermal of microRNA, isolation of microRNA and small interfering RNA. So this is the method. Now I'll see purification technology. Nucleic acid purification technologies. There are different kind of nucleic acid purification technologies. Storage of eluted DNA or RNA can be done for preserving its integrity, basically stored in -20 and -80. Purification using small scale, small automated system offer benefits, as you can see. Now I'll see purification manual. Low investment and suitable scalability are attractive. Purification using the automated 96 well, as you can see in the figure. So these are advantages. And purification using manual 96 well. How to concentrate dilute nucleic acids. So basically dilute nucleic acids can be concentrated. Water molecules can be removed from dilute aqueous nucleic acid solution by extraction, ice cold ethanol precipitation, direct evaporation by resuspension. The procedure is useful for reducing the impurities.